Yo, what's up, guys? Today we'll be talking about carbohydrate metabolism, but this carbohydrate metabolism is not an in-depth one because this carbohydrate metabolism is just in relation to neurobiochemistry. Okay, telling you how energy is being supplied to the nervous system. Okay, that's the biochemistry, the carbohydrate metabolism that we'll be discussing here. Now we see that the brain contains a very small store of glucose and glycogen, okay, compared with other tissues. Now brain tissues also contains intermediate of glucose metabolism because of blood-brain barrier. Now because of blood-brain barrier, most of the other carbohydrates are absent or present in negligible amounts in the brain. Okay, and also the nervous tissue oxidizes what glucose and releases energy mainly in three interrelated what phases. That's the glycolysis, the Krebs cycle, and the electron transport chain. Okay, I just brought this image here. You see the image of glucose, fructose, and mannose. Okay, now glycolysis may take place aerobically or anaerobically. The Meyerhof scheme of glycolysis occurs mainly in the central nervous system, and alternative pathways do occur, but they have shown to be of little importance. Right now, the last two phases, during which pyruvate is oxidized to carbon dioxide and water, supply the major portion of energy for cerebral function. Okay, now a minimal amount of glucose is metabolized by cerebral tissue via the hexose monophosphate pathway, mainly for the production of what reduced NADP. Right now, this is required for the synthesis of fatty acid and steroids. Okay, um, proceed. This is the structure of glucose. I just brought, since we are talking about glucose and energy, I just brought this structure so that I could remind you of the main carbohydrate metabolism. All right, I will be making videos on the main carbohydrate metabolism where we have all those many steps. All right, we're making videos on that. Now, we say that in the absence of adequate supply of glucose to the brain, only a few um okay, only a few substrates of brain energy. Sorry, in the absence of supply of glucose to the brain, only a few carbohydrates have shown to be of act as substrate of brain energy metabolism. Okay, now compounds such as maltose, fructose, galactose, and intermediates such as lactate, pyruvate, and glyceraldehyde were shown to do so only after their conversion to glucose elsewhere in the body. Thus, these compounds they act by rising the blood glucose level. However, the brain can utilize mannose directly and rapidly. Okay. Now, in the absence of glucose, to restore or maintain normal function. In other words, mannose can enter directly into glycolytic pathway of the brain tissue without raising blood glucose level. Okay. Now we say that mannose like glucose can easily cross the blood-brain barrier and can be converted to mannose 6-phosphate. Okay, this is where this image comes in place. This is mannose 6-phosphate, right? Learn how to draw it. Now this is done by the enzyme hexokinase, right? Now the rate of this reaction is about equal to the rate of conversion of glucose to what glucose 6-phosphate by the same enzyme. This is what glucose. This is glucose 6-phosphate, all right? Glucose 6-phosphate. Now the enzyme phosphomannose isomerase, an active enzyme in the brain tissue, converts what mannose 6-phosphate to fructose 6-phosphate, which then enters the glycolytic pathway. And note that the mannose is not normally present in blood in any appreciable amount. Okay, so this is what the fructose 6-phosphate that we are talking about. I hope this image makes sense to you. All right, now you could learn to draw these three structures that I cycled out. Okay, so that I could know where at which carbon atom they differ at. All right, and how they differ. All right, so see you guys in the next tutorial, and bye for now, guys.